All right, let's talk about viruses. Viruses are not considered a life form by most scientists, but a parasitic chemical, and that's because they are missing several characteristics that are present in all living things. They can't reproduce without using an actual living cell. They don't have any metabolic activity of their own. They can't make any proteins on their own. They don't synthesize anything on their own without being inside of a cell and using the cell's machinery to make its proteins. They do, however, have heredity. They have either a DNA or an RNA genome, and because of that, mutations occur, and that means they can evolve. A protein is a protein sheath called a capsid is surrounding the genome of every virus, and those are the two basic things that make something a virus. It has a DNA or RNA genome, and it has a protein capsid surrounding that genome. Now, some viruses have an extra third component, and that is a fatty envelope. This is often made of a phospholipid bilayer that they steal from the cells they infect on the way out. Viruses are also very small. Almost all viruses are smaller than the smallest known cells, although we'll talk about some really big viruses that have been discovered more recently. This is a nice electron micrograph of some herpes virus in comparison to the size of a human cell. So we can only see a tiny corner of this human cell with these tiny little viruses surrounding it. Viruses were discovered indirectly without being seen. They were discovered by their ability to transmit infection, but being smaller than the smallest cells, they could pass through filters that could filter out bacteria. The first virus to be demonstrated to exist was the tobacco mosaic virus. The tobacco mosaic virus causes a disease in tobacco leaves that leads to spotting and wilting of the tobacco plants and gives them the dead spots, and the yellowed spots give it kind of a mosaic appearance, and that's why it's called tobacco mosaic virus. So it was hypothesized that whatever was causing this infection was smaller than bacteria. And the way that they demonstrated that was by taking an infected plant, passing it through a ceramic filter that could filter out all the known bacteria, the smallest known bacteria. So it had a pore size smaller than 1 micrometer. And then they would take the liquid that was able to pass through that filter and put it on a healthy plant, and they would get sick. So the infectious agent was very tiny and not visible to the microscopes of the time. So the microscopes of the time could not see things that were much smaller than 1 micrometer, because about 0.4 micrometers is the smallest wavelength of visible light. So that's about as small as you can see with a light microscope. Electron microscopes, though, were invented in the beginning of the 20th century, and that led to actually seeing viruses for the first time. Viral genomes can have DNA or RNA, but that RNA or DNA can also be in the form of a double or a single strand. So there are kind of 4 big categories of viruses: double or single-stranded DNA or double or single-stranded RNA for their genomes. And that's going to make a difference to how that virus particle is going to replicate inside of cells, whether it's a DNA or an RNA virus. The capsids are made of proteins, and the genes for those capsid proteins, because remember, if it's a protein, the way that a protein gets made is there has to be a gene and then a messenger RNA, and then that goes to the ribosome and makes the protein. So those viral capsid proteins are encoded by genes in the viral genome. So here, and they vary quite a bit. So here's just 4 examples of very different viral capsids. This is the tobacco mosaic virus, this first one here on the left, and it has a very simple structure. It has a single protein in its capsid that's repeated over and over again hundreds of times, and surrounding its RNA core. So it's quite a simple virus. Adenoviruses can infect humans. These are often used in genetic engineering because they cause very mild illnesses in humans, mild respiratory illnesses and cold-like symptoms, but they have a DNA genome and you can insert genes of your choice into them and infect people with them experimentally. So this is one of the viruses that's being used experimentally to try to develop a vaccine to the coronavirus by adding coronavirus genes to the genome and getting it to express proteins on its surface that will activate an immune response against coronavirus. Hopefully, it's one of the ones that's in development. Influenza viruses have that additional phospholipid membranous envelope. So they have genome, protein capsid, and then the outer membrane that they steal from the cells that they infect on the way out. And this is what makes them vulnerable to alcohol-based hand sanitizers, which we'll talk more about. Coronaviruses as well have this outer membranous layer on them. The most complex capsids are found in bacteriophages. These are viruses that attack bacteria. They have been co-evolving with bacteria for perhaps 3 billion years. They have several different types of protein in their capsid, and the protein itself does not enter the bacterial cell. Instead, they inject their DNA into the bacterial cell, and the protein capsid actually remains on the outside. And they do look very bizarre, sort of like the lunar lander or something, and not really like something biological, but nevertheless there's an electron microscope image of one. So that fatty outer envelope that some viruses have, these are often found in viruses that are found in animals. The virus has to steal this membrane from the cells that it infects. So viruses that have these kind of envelopes wouldn't be found to infect bacteria or even plants because of the cell wall that's in the way. Colds versus flu viruses. Okay, so flu viruses, as I already mentioned, and coronaviruses have that fatty outer membranous layer. So that means they are vulnerable to alcohol-based hand sanitizers because while most of these hand sanitizers use ethanol, but some also use isopropyl alcohol, which is rubbing alcohol, both of those can dissolve lipids. So when you have something with a fatty outer layer, like a flu virus or a coronavirus, and you put alcohol-based hand sanitizer on it, it actually breaks up that outer layer. However, it's not as effective as soap. So if you really want to get rid of any viruses or bacteria on your hands, you've got to wash your hands. Cold viruses don't have that fatty outer layer. They are completely invulnerable to alcohol-based hand sanitizer. In fact, alcohol is used to preserve them. So if you're preserving cold viruses in the lab and you're going to freeze them, you first put them in ethanol and then you put them in the freezer and they will remain active. So alcohol has no effect. So if you're using hand sanitizer to try to prevent the common cold, it's not only is it useless, it probably spreads them out into a thinner layer so that you have greater chance of infecting others with them. So wash your hands is the moral of that story. Phages or bacteriophages are just viruses. They're viruses that attack only bacteria, however. They can't attack our cells. They attach to the cell wall on the outside of the bacterial cell and then inject their DNA into the cell. They have very complex capsids and multiple proteins that the capsid is made out of. This is a nice electron micrograph image here with lots of little phages attacking this one bacterial cell, which is most likely an E. coli. So viruses have a particular host range, and the host range is the number of species that it can successfully replicate in. So for the flu, for influenza, it has a fairly broad host range, and it can infect multiple primates, different species of birds, including chickens and ducks, and also domestic pigs. So it has a very large host range, and other viruses have a very small host range, like measles is a human-specific virus. It might be able to infect closely related species like chimpanzees, but it's not going to infect your pets or your chickens. And even within the species that a virus infects, there's only specific tissues that it can infect. So viruses bind to proteins on the surface, and only certain tissues in that host are going to have the right proteins on the surface for it to bind. For example, cold viruses can bind to proteins that are found usually just in mucous membranes. So that means cold viruses can infect your nasal cavity, your sinuses, sometimes your small intestine, because that's the cells that have the right surface protein. But a cold virus is not going to infect your liver or your brain because those tissues don't have the right proteins on the surface for them to infect the cell. So once a virus gets inside of a cell, it breaks apart, and its genome then becomes available for replication. So the viral genome is going to be replicated, its proteins are going to be made, it's going to use all the host's ribosomes, enzymes, building blocks, tRNAs, amino acids, ATP. Viruses don't even have any way to get energy or use energy. It's completely the host cell that is providing all of those things. Occasionally, viruses will bring an enzyme with them that a host cell would not have, like reverse transcriptase in the case of HIV, or an RNA to RNA transcriptase, which some RNA viruses have. No living cell would have that enzyme. So virus particles self-assemble generally. So once the new genomes and the capsid proteins are replicated, the virus particles will self-assemble. And sometimes that alone is what kills the cell. It's just the total hijacking of all the cell's manufacturing for making new virus particles that causes the cell to rupture. So here's a typical viral infection here. This example is a virus with a DNA core. So viruses have proteins on the surface. They're either the capsid proteins themselves, or if it's an enveloped protein, then it's proteins that are sticking out of that membranous envelope. And those proteins match to something on the surface of cells, very often receptors. The receptors are there for something else. The receptors are there as hormone receptors or for some kind of intracellular signaling in the organism. And the virus just happens to match that same receptor. And that's how it gets into the cell. So, but it has to have something to stick to, or it can't get into the cell. So once the viral genome is in the cytoplasm of the cell, if it's a DNA virus, like this example here, that DNA can be a template for both DNA replication to make new copies of the viral genome, and also for RNA transcription, because there are genes in that viral genome for making the capsid proteins. So those genes will be expressed just like any other gene, and RNA copy will be made, and then that will go to the ribosomes, and the capsid proteins will be made. And then the viral particles will self-assemble, and then they may be exuded from the cell, or they might rupture the cell, and that's how they get out of the cell. Phages are very well understood because possibly the best understood cell is not a human cell, but an E. coli cell, and the viruses that attack that cell are phages. These viruses have two basic life cycles, one that kills the cell immediately and one that will allow the cell to replicate. So our phage here infects our bacterial cell, and it's going to do one of two things. Either it's going to trigger the breakdown of the host genome and replicate and lyse the cell and release thousands of viral particles immediately, or because these are all phages or DNA viruses, it can just insert itself into the genome and then just sit there in the genome while the cell divides and divides and divides, and then pop out later. And generally they are triggered to go back into the lytic cycle by some environmental condition, or by if the cell is not dividing, if the food supply is in short supply for the cell, they will be triggered to switch back to the lytic cycle. Viruses that attack our cells, some of them can do the same thing, can insert themselves into the genome and be carried along by cell replication. They insert themselves randomly, so sometimes this is how mutation occurs, that a virus inserts itself into the middle of a coding region of a gene and disrupts its function. Not all viruses can do this, only some viruses can do this. So we'll go on next and talk about some specific viruses.